Okay, good afternoon. I hope everybody enjoyed his lunch. We're going to continue our afternoon session. And the first talk in this session will be by Janak Raj Joshi from Nepal. Janak has graduated his BSc degree in agriculture from the Institute of Agriculture and Animal Science in Tribhuvan University, Nepal. Janak, please.
Good afternoon, everybody. Excuse me.
Sorry.
Good afternoon, everybody. I am Janak from Nepal, and the topic of my research exercise is examining the sensitivity of ROS-related mutants to various stresses, which was done under the supervision of Dr. Friedman in Department of Post-Harvest Science, Vulcani Center. Reactive oxygen species, abbreviated as ROS, they are chemically reactive molecules containing oxygen, which are produced continuously as a byproduct of aerobic metabolism. And this reactive oxygen species, they are produced from triplet oxygen, which is the ground state oxygen, either by energy transfer or univalent reduction of this triplet oxygen. Here we can see the triplet oxygen, on getting energy, converts into singlet oxygen, which is one of the member of reactive oxygen species. Similarly, when it gets reduced by getting electron, it gets converted into superoxide radical ion, which is another member of reactive oxygen species, and which on protonation gives perhydroxyl radical. Similarly, on further reduction and protonation, we get several species of reactive oxygen species. This is our reactive, so we can say that this triplet oxygen might be the foundation of the members of reactive oxygen species. Reactive oxygen species, they are produced in different cellular compartments, especially in chloroplast during photosynthesis, in peroxisomes during photorespiration, in mitochondria during respiration, and in apoplast during growth and development by the action of oxidases and peroxidases. But under normal conditions, the level of reactive oxygen species in each of these compartments is maintained at their optimum level by the action of antioxidants or ROS scavengers. They are maintained at their optimum level. But under abiotic stress or attack of pathogen due to the activity of hormones and due to growth and development, the ROS gets accumulated in different cellular compartments. And this accumulation of ROS in different cellular compartments might be either be due to overproduction of ROS in response to these stresses, or might be the reduced activity of the scavengers or the antioxidants, or combination of both. So when ROS gets accumulated in different cellular compartments, then it acts as a signaling molecule, and the signaling molecule and the signaling is specified by the location of the reactive oxygen species, in which compartment they are located, or the concentration of this reactive oxygen species in the specific compartments, or the duration of the reactive oxygen species in the specific compartments. Because these reactive oxygen species are highly reactive and not stable, so the duration also determines the specificity of the ROS. So when this reactive oxygen species gets accumulated, then the signal is perceived, which is responded either as a protective role or as a destructive role by initiating the cell death. Gazev in 2006, he reported 32 genes that were induced under various abiotic stresses. Among the genes, function of some of the genes were still unknown. Similarly, Rosenwasser in 2011, he compiled 44 transcription factors that were induced in various oxidative stresses. Since these genes and transcription factors, they all were induced in abiotic stresses, so we expect that they play a role in multiple stresses. Moreover, the role of these genes and transcription factors are still unknown in various abiotic stresses. We don't know the clear function of these genes and transcription factors in various abiotic stresses. And these are the oxidative stresses that were implied for studying the expressivity of the genes and transcription factors that I mentioned earlier, like using flu mutant or knockdown of superoxide dismutase or alternative oxidases or ozone fumigation using methyl violation. These all were the agents for inducing oxidative stress. And this is the list of genes from Gazeb that were induced during oxidative stress. And these genes were also induced during dark-induced senescence. And the values here, which are shaded under red, they indicates the upregulation of the genes by at least 2-folds on treating with extended darkness. From the list, I selected the 3 genes. 2 of them are expressed protein. The function of these genes are called expressed protein because the function of this protein is still unknown. So they are nomenclatured as expressed protein. So I selected 2 of the expressed protein and 1 tall B protein related. This is also a gene. And this is the list of transcription factors that were induced under various implied oxidative stresses. These transcription factors were also induced in dark-induced senescence. The cluster of genes that are shaded under red, these are the upregulated gene under dark-induced senescence by at least 2-folds. And these are the cluster of transcription factors that were downregulated under dark-induced senescence. So from the list of these transcription factors, I selected 4 transcription factors randomly. So the objective of my study is to elucidate the role of these ROS-related genes and transcription factors in various abiotic stresses. There are various abiotic stresses that we studied, among which the sorbitol, salt, methyl violation, heat, and UV. The effect of these abiotic stresses was studied on the root growth of young seedlings, whereas the abiotic stress of extended darkness was studied on 5-week-old bolted plants by examining the chlorophyll content and leaf cell death. So, to study the effect of salt, sorbitol, and methyl violation, we grew the sterilized seeds of Arabidopsis in MS plates, supplemented with NaCl, sorbitol, and methyl violation, among which we took salt because it induces salinity stress, we took sorbitol to induce osmotic stress, and we took methyl violation to induce oxidative stress. So the seeds after sterilization were planted in the MS plates and they were kept in cold chamber for cold treatment. Since Arabidopsis seeds require at least 48 hours of cold treatment to induce germination, we just kept them in cold for at least 3 days in 4 degrees centigrade. After that, they were transferred to growth chamber and the plates were positioned vertically so that root would grow straight downwards and it would be easier for further measurements. Then root lengths were measurement 6 days after they were transferred to growth chamber. For studying the effect of heat and UV treatment, the seeds were planted in MS plates not supplemented with any chemicals that induce stress, just the MS medium. Then, as I did earlier, the cold treatment was given for 3 days and they were transferred to growth chamber. And immediately after germination of the seeds, we imposed our assay of heat treatment, and our assay was 50 degrees centigrade for 30 minutes. And the UV treatment was given under the UV light for just 5 minutes. And they were again transferred to growth chamber after the treatment, and root lengths were measured 4 days after treatment. So combiningly it will be 6 days, because we took the data 4 days here and they already stayed in growth chamber for 2 days. So we took data after 6 days. And for dark treatment, we used the 5 weeks old bolted Arabidopsis plants. So we need to grow them in pots. So we use the peat mix to grow as a we use peat mix as a growth media for growing Arabidopsis plants. We put few seeds and then again cold treat them to induce the germination. Then they were transferred to growth chamber, and after 5 weeks we detached the rosette from the whole plant and kept it under extended darkness for 3 days. Now the treated rosettes, from the treated rosettes we took the leaf disc of 5 millimeter from young leaves as well as old leaves, and the leaf discs were used to determine the chlorophyll content in them and the cell death in them. And the chlorophyll content was determined by using spectrophotometer, and the cell death was measured by using FL600 microplate. This is the model of one of the machine as a spectrophotometer, by following Evans blue staining. So on treating the mutants we selected under sorbitol stress that creates osmotic stress, we found that the wild type itself is little bit sensitive to the supplied osmotic stress. Here these are the values representing the ratio of the root length of treatment versus control. So if anything below 1 means it is susceptible to the stress, and anything above 1 means it is resistant to the supplied osmotic stress. So here wild type is itself little bit sensitive to the supplied osmotic stress, and the mutants AP2 transcription factor, Tolby protein related, Rocky53 and NAM, these 4 mutants, they resemble in phenotype with the wild type, whereas these 2 expressed proteins, the mutant of these 2 expressed protein and Rocky25, they were protecting against the stress because they have enhanced root growth, which means that these genes, these 2 genes and this transcription factor, they are involved in enhancing the sensitivity towards osmotic stress, because in their absence they are protecting. So when they will be present or when the genes will be expressed means they might reduce the root length, which means increase the sensitivity towards osmotic stress. So we can predict that these 2 expressed protein and this transcription factor might be involved in increasing the sensitivity of the plant towards osmotic stress. And on treating the mutants with salt treatment, the wild type was indifferent. We didn't see any effect of this salinity level on wild type. And these 2 expressed proteins and this AP2 transcription factor, they also resemble to wild type. We didn't find significant difference between wild type and these 3 mutants, whereas these 4 mutants with 1 gene, Tolby protein related, and these 3 transcription factors, they were sensitive to the supplied salinity stress, which means that these transcription factors and gene might be involved in providing or increasing resistance of the plant towards salinity stress, because in the absence of these genes these are sensitive, means when they are expressed, they might enhance tolerance towards salinity stress. Similarly, on treating the mutants with methyl biologen, that is the source for creating oxidative stress in the plants, wild type itself is slightly sensitive to the supplied stress, whereas this mutant, the expressed protein 13, the tall B protein related, and Rocky 25. These three mutants were sensitive, much more sensitive as compared to wild type under oxidative stress. So we can say that these three genes, these two genes and this one transcription factor might be involved in providing resistance towards oxidative stress, whereas this Rocky 25, this mutant showed slightly protective role against the oxidative stress. So we can say that this Rocky 53, this transcription factor might be involved in enhancing the sensitivity of the plants towards oxidative stress, because on the absence of this gene, the plant is sensitive. On treating the mutants under heat at 50 degrees centigrade for 30 minutes, wild type was sensitive, and the same trend is followed by this expressed protein 13 and Rocky 53. Whereas the other mutants of AP2 transcription factor, tall B, NAM and Rocky 25, these all were protective. These mutants were protective against heat stress, which means the genes and transcription factor themselves, they enhance the sensitivity of the plant towards heat stress, because in their absence they are protecting, means when they are present, then they will increase the sensitivity of the plant towards heat stress. But the interesting thing we found here is the expressed protein. What happened in this? When we treated the mutant with heat, all plant died. They turned brown with no further growth of roots. So we can expect that this gene might be involved in enhancing tolerance against heat stress. As I mentioned earlier, this expressed protein, it was nomenclatured as expressed protein because there is no known function of this gene. So we can predict that this gene might be involved in providing resistance against the effect of high temperatures. This is the expressed protein that I showed earlier, which on non-treatment have luxurious growth of roots, as you can see here, even in wild type. But when we treated it with heat, then you can see the plants. Almost all plants were turned into brown and they started dying. So we can just predict that this gene might be involved in providing resistance towards heat treatment. On treating the mutants with UV light, we found that the wild type itself is sensitive towards the UV treatment, and this expressed protein resembles to wild type, so does the Rocky 53, tall B and Rocky 25, whereas AP2 transcription factor and NAM transcription factor, these both mutants were protecting the plants against UV stress, which means when these genes are expressed themselves, they will increase the sensitivity of the plants towards UV stress. But look at here, the mutant of expressed protein, it is much more highly sensitive as compared to wild type. So we can say that this gene might be involved in enhancing the tolerance of the plants against UV stress. This is the mutant expressed protein which was not treated with UV light, we can see here luxurious growth of roots in normal condition, and so does the wild type when they are not treated with the UV light. But on treating the mutants with a mutant and the wild type with UV light, we found much more reduction in root length. You can see here some of these plants died because of the treatment, and the other they survived but with no further significant growth of roots. You can see here clearly, but in wild type there is reduction of root growth, but not as sensitive as expressed protein. So we can say that this expressed protein, the function of which is also unknown, might be involved in enhancing the tolerance against UV light, against UV stress. These are the results that we got after treating the mutants under extended darkness for 3 days. The rosettes, as I mentioned earlier, they were treated in darkness for continuously for 3 days, and we measured the chlorophyll content of the treated leaves. And what we got is the wild type with the chlorophyll content at the same level in old leaves and in the young leaves. But when we compare the mutants with this wild type, this expressed protein 13, AP2 transcription factor, TOL-B, and ROCK25, these all mutants, they have higher chlorophyll content in their younger leaves, which indicates that when the gene are expressed themselves, when the gene and transcription factors are expressed themselves, it means we will have reduced level of chlorophyll content in them. Reduced level of chlorophyll content means it is the initial stage for senescence. The senescence starts with the degradation of chlorophyll. So we can predict that these genes and transcription factors, these 4 genes and transcription factors, might be involved in enhancing the senescence process upon their expression. Whereas this non-transcription factor, this has significantly lower level of chlorophyll content as compared to the wild type, which means this non-transcription factor itself enhances the tolerance towards dark-induced senescence, because in its absence we have low chlorophyll content, means when it will be present it might have higher chlorophyll content. It means it is prohibiting the plant from degrading the chlorophyll upon dark treatment. Similarly, on treating the mutants with extended darkness, we measure the level of cell death, which is the final consequence of senescence process. We found this expressed protein with lower level of cell death, and so does the AP2 transcription factors, younger leaves of TOL-B protein related, ROCK53, and non-transcription factor. They all have lower level of cell death in the absence of these genes and transcription factors. It means when they will express themselves, they will have higher level of cell death, which means they induce senescence process. So we can say that these genes might be involved in inducing the senescence process. Whereas this ROCK25, even in younger leaves we observed higher level of cell death. So in absence of this transcription factor, we expect to have lower level of cell death, which means it reduces the activity of extended darkness on senescence. So we might predict that this gene might be involved in protecting the plants against senescence. So this is the compiled result of the treating the plants under different abiotic stresses, in which the red color indicates for the protective role, and green represents for the sensitiveness. This is just the compiled result that I showed you earlier, and black represents it is indifferent from the wild type. So concluding the expressed protein, which is the protein of unknown function, might be involved in protecting plants from high temperature effects, because the mutant itself was highly sensitive towards heat stress. Similarly, the expressed protein, which is the hypothetical protein, might be involved in mitigating the negative consequences of oxidative stress, which was created by UV light and methyl violazine. And this mutant was also found sensitive to the methyl violazine treatment of 0.1 micromolar by Luha et al. In 2008, which supports our result. And this AP2 transcription factors was highly sensitive to heat and UV stress. As we showed earlier, the mutant itself was protective. So it might be involved in triggering the activities of senescence associated genes also. TOL-V, non-transcription factor, ROCK25 and ROCK53, they were expected to enhance tolerance against salinity stress. And our results were analog to findings of Hu et al. And Jiang and Diholos in 2009. What they found is they overexpressed the ROCK25 and non-transcription factor. The overexpression lines of these two transcription factors, they enhance the tolerance in salinity stress, which is in parallel with our results. Similarly, ROCK53 might assist in enhancing the senescence process. And our results were similar with Mia et al. In 2004. What we found is overexpressing the ROCK53 is induced the senescence process. Similarly, Rivero and Luha, they found stress-specific response on the expressivity of the specific expressed protein. They worked only on the expressed proteins, the function of which is unknown, and they found that the response is specific to the type of stress plant is bearing. So we can conclude that not all the genes are acting equally to protect against different stresses. Means the genes, they are specific to the type of stress that they are experiencing. So ROS-related genes and transcription factors are specific to the type of stress experienced by the plant, though they are induced in several stresses. Thank you.
Thank you, Janak. Yes, Sarai, take the mic.
Thank you, Janak, for your nice presentation. I have a series of questions, actually. Okay. You used 2 expressed protein and 4 transcriptional factors out of the lists, right?
Yes.
And what was the basis for you to choose those? The other, use sodium chloride as salinity stress and sorbitol for osmotic.
Yes.
But salinity stress also causes osmotic, especially for short experiments like you. The first thing is osmotic problem due to salinity.
Yes.
I mean, they may. And the other, when you.
One at a time. Let him answer one by one.
Okay.
One by one. First one.
I selected the mutants just, I can say randomly. I selected them randomly, but the randomly doesn't work because I should have the mutants. I should have the mutants in my lab. So I just selected randomly, just 4 or 5, and the thing that coincide with the mutants availability in my lab, I choose them. And salinity stress also induces osmotic stress, but it's not, they are not the same. The salinity stress is different from the osmotic stress.
What is that?
The osmotic stress, when you use salt, then we get ionization of the salt. So these ions also have effect on the growth of roots, but in osmotic stress, we don't get ionization. So this makes the difference in both of the stresses.
Okay. to classify them as susceptible and resistant, use ratio, and those who have less than 1, you classified as susceptible.
They are susceptible, yes.
What is your base to put 1 as a demarcation?
The wild type. I
Okay.
compared everything with the wild type.
With the wild type, this is a,
No,
this
no,
is
you didn't. You compared treatment to control.
this is also the question that I wanted to ask.
Here,
The relative values are
Yes.
relative.
they are the ratios of treatment versus control.
versus control of the
Yes.
wild type.
No,
or
no, no,
each
of
one
themselves.
versus its own control.
Each one versus its own of its own control. Each one versus its own control.
So if you got the growth at treatment is higher than the control, of course you will get more than 1 value.
Yes, I will get
So-
more than 1.
So that infers it's tolerant.
Yes, that infers that the mutant is tolerant itself, but not the gene. The gene is the gene when expressed, plant will be more sensitive. But in absence of this gene, this is a mutant. So this is a mutant means this gene is not present in this plant.
Okay. And the other, you took only root lengths, right?
Yes.
And those who have longer root lengths, I mean under treatment, then you'll.
They are protected, means
They are
the
protected.
mutant is protected.
But sometimes it's misleading to take root lengths alone because you have to also consider the above shoot growth because it may be biomass partitioning problem. If under stress, that line, I mean the biomass partitioning will be toward the root, then it will have more root growth than the shoot part. Even sometimes those who have less root growth under treatment under these stresses may be preferred than those who have longer root lengths, depending on their above ground part growth.
Yes, but we took only the parameter of root length. We didn't measure any above biomasses.
Yeah, I
We
mean,
just took the
but
root
taking
length.
root lengths only, you can't say that they are resistant or tolerant.
Yeah, but the main thing that guides are the roots. On the basis of root, the plant develops.
Yeah,
Root are also the primary things, so I just took the roots.
It would have been better if you also considered the.
Yes, it would have been better, but I took only roots in this experiment. So maybe some should.
Did you observe any cases in which the results that you got for the root,
Yes.
that you got just the impression from visualizing the treatments, the plants, the plates or whatever, that the results you got with the roots are not really representing also the shoot.
Oh, but since I grew them in MS medium, the nutrient was not a problem. So I didn't took the data for the shoots,
No, I mean,
but
it
just
wa-
on visualization it was okay because there was not a problem.
Did you observe that normally when root is short, also the shoot was short, or there were cases in which the root was short, but the shoot was longer or shorter, that there was
I didn't
variation,
observe that
it
because
was not correlated, that the shoot length
I
was
didn't
not
observe that.
correlated with the root.
I didn't observe that because that was not the part of my parameter, so I just concentrated on the root length.
One last question. For heat treatment, you export them only for 30 minutes.
Yes.
Is that the standard? I mean,
No.
is it short?
No, the heat treatment, this is not the only one treatment I did. I did it even for 5 minutes, but the significant results I got after treating with the 30 minutes. So I just put this data here.
No. Okay, you started from 5 minutes.
Yes.
Even I'm
and
saying
the
that
result
this
was
is
not
short.
significant. Not
I'm
at
saying
all,
that this is
nothing.
small duration of exposing.
Yeah, even
exposing.
it is a small duration, but it seems very effective. So I just took this assay.
I mean, you can take, but you can't infer that they are resistant or.
But it's 50 degrees.
Yeah, but 30 minutes is, I think, from the lifetime of. I
No,
don't
it
know,
means
30
in this short period of time, if they are experiencing some sort of differences, means on longer treatment, is for sure they will have differential response.
Yeah, I think it's a very valid I don't know, 42 degrees for I don't know, for several days, then it does not, it's not necessarily the same, you will not get the same result if you will expose them for longer time, for lower temperature, to simulate real life, right?
Yes.
It's a valid criticism.
You find some genes which can protect the plants from oxidative stress and some other stress.
Yes.
Yeah. And do you know that the genes are found in some other plants also? So
Yes.
that, yeah, which,
The genes, since Arabidopsis is a model plant, we just worked in it, and these genes, if we know the function or the role of the genes in abiotic stresses, then we can go for the further reading in the other plants, and we can imply it.
do the same gene, they are discovered in some other plants now?
I don't know that.
Okay, if it is discovered in some other plants now, then you can overexpress that gene and prevent from the oxidative stress and some other stress.
Yes.
Okay, thank you.
So did you just do replicates with the same amount of sodium chloride and sorbitol?
Yes.
Or do you think
No.
if you have used different amounts, you would have observed some other
I
stuff?
observed from the range of 0 millimolar through 300 millimolar of salt and sorbitol. And I found the significant results on 100 millimolar of salt. So the 100 millimolar of salt is equivalent to 200 millimolar of sorbitol. So I took 100 millimolar of salt and 200 millimolar of sorbitol, though I took the value in a range from 0 millimolar to 300 millimolar.
Is equivalent in what respect? The osmotic effect of
Yes.
the salt and the
Yes.
sorbitol?
Yes.
Yeah, thank you. My question is on the expression of the ROS. I really want to believe the expression of ROS have an advantage, because in your introduction you said that in the normal circumstances, that is naturally, it's expressed. The level of expression is to the level that it will not accumulate in the
Yes.
in the plants. And then you went further to explain that during a pathogen attack,
Can I-
and then
They accumulate. I don't say they never accumulate. They accumulate, but the level of their toxicity, it can't be seen. They are not toxic. The
Okay.
toxic level is minimized by the action of scavengers or the antioxidants.
however, during a pathogen attack
Yes.
or abiotic stress,
Yes.
the level of accumulation is high and it can affect the plant. Is that right?
Yes.
All right. So I really want to believe that the expression of ROS, especially during the pathogen attack, have an advantage. Do you believe that?
Yes, they have advantage.
Okay. Then my question is, during your studies, did you come across some genes that can be improved to decrease the levels of the ROS expression, so that in case of pathogen attack or abiotic stress, that the plant have to express ROS to protect itself? Did you come across any gene of that sort?
Look, this ROS previously, in the earlier studies or previously, they have a concept that ROS they are just damaging. They are harmful molecules. But now the present studies, many studies have shown that they are not only the harmful one. They have some protective roles also. As you mentioned earlier, in pathogen attack, they protect plant. But the cell itself is damaged. Even then, the cell, for cell, it's a damaging role. But under stresses, since it acts as a signaling molecule, so when it signals the plant, then there are some genes that provides resistance of the plant towards the stresses. So the signal might be protective by inducing the genes that protect the plants from specific stresses. So there might be gene.
Yes, my question is on this diagram. I want to know how does the antioxidant activities reduces overproduction of ROX?
No, I didn't say that overproduction of this reduces this. This is not the thing I told.
Yes, but if we have accumulation of the ROS, it has oxidative damage to the cell, right?
Yes.
So how does the AOX, I mean the antioxidant activity, I mean reduce the...
They are reduced by these factors.
Okay.
This is affecting this and this. This is not affecting this. This scavenging activity and production, these are affected by this independently.
Now let's assume that the abiotic stress doesn't, I mean, it's not a problem, okay? But how does the antioxidant, okay, if there's too much of the ROX produced, how's the antioxidant activity, let's say vitamin C or something like that, VC, vitamin C is an antioxidant, right? So how does this antioxidant reduce the overproduction overproduction of the ROS if...
It depends upon the specific antioxidants. Some, they don't allow the production of ROS. There is a pathway, so some reactions by reduction, as I showed earlier. Some stops there. And some, in some cases, there is one superoxide dismutase. In some cases, they just convert the radicals into water, so they don't allow the production of this. And in some cases, they produce the proteins for quick recovery after the damage. So there is not just a single pathway or a single mechanism. It can be, the damage can be mitigated either directly or indirectly, or before production of ROS or after production of ROS. It can be anyway.
Okay, thank you very much, Janak.